All right, thank you, Hollis. Can you see my screen?
Yes, we can.
And
right.
I sound good? Excellent.
Yep.
Well, thanks for that introduction, Hollis, and really thank you and Brianne for organizing all this and all the RCN workshops. I've been a participant and a fan since the beginning, since we met in Shepherdstown, I think 4 years ago now, and I'm firmly behind this process and the need for this. And I think it's really exciting to have this little microcosm today, this kind of case study that we're going to be talking about for BLM as the potential, way to look at how we're going to do this nationwide. So, so I'm here to at first at least cover an overview of the Bureau of Land Management in a very general sense, and then kind of zooming into our pollinator and bee monitoring world to give you all some context, because I think many of you probably are familiar with the BLM. Maybe some of you on the eastern part of the US are less familiar with us. We're not as well known as some of our sister agencies like NPS or refuges or even the Forest Service that are managing other large swaths of land. But I always like to promote us as being a very important land manager in the United States and important for species and habitat conservation. And I think this today just helps further that. A little bit briefly on myself, I'm the Wildlife and Threatened Endangered Species Program Lead for BLM in Alaska, but I also help support BLM nationally in pollinators and bee monitoring. So that's kind of why I'm taking on this role today. And I've been working on pollinator conservation for probably 15, 17 years now. And it's just been exciting to see the growth and excitement in, nationally and beyond in the recognition of the value of pollinators and the need for their conservation, and ultimately the need to monitor them to understand the situation. So let me jump into this presentation here. So again, we're going to start out on the really kind of big picture with the BLM to give you all some context, and then zoom down to some of the more specifics in the bee world for us. So we are the largest land manager in the United States. We manage more than 10% of the total surface of the United States, but we also manage about 30% of the subsurface mineral resources in the US. We are known for our multiple use and sustained yield mission to sustain the health, diversity, and productivity of public lands for the use and enjoyment of present and future generations. So we do have a challenging but I think worthwhile mission. Our land is not just set aside for conservation. We do have other uses on the land, and we have to balance those uses on a landscape scale. There are 6 principal or major uses defined in the Federal Land Policy and Management Act, FLPMA, and fish and wildlife is one of those 6. You can see the others there: livestock, minerals, right-of-way, recreation, and timber. Over on the right too, you probably already noticed we have a map where you can see in yellow the BLM lands across the country, as well as the cities where our offices are. So if you're working in a state or more locally, you can kind of get an idea of where you need to reach out to our local folks. I'll share this map again, I think, at the end of the presentation. So our BLM policy is to manage habitat with an emphasis on ecosystems to ensure self-sustaining populations and a natural abundance and diversity of wildlife, fish, and plant resources on the public lands. So that gives us a lot of push to monitor not just sage grouse, not just mule deer, not just the more well-known charismatic large species, although I argue that bees are charismatic. But it really does push us to understand and manage and monitor the full natural abundance and diversity of our resources, including bees and invertebrates and a lot of things that get maybe less attention but deserve it. One of the things that I really love to highlight is that BLM manages some of the most species-rich and abundant bee habitats in the United States. And you can see the 2 maps here, where you can see these hot spots in the West, especially the Southwest, for richness and abundance. And those are areas, if you remember the last map, where we have fairly extensive BLM managed lands. So again, not only are we the largest land manager, but we do manage some of the most important habitat for bees. We are currently near final, but this is still a draft in our BLM pollinator strategy. And goal number 1 is an inventory of BLM lands and identify management needs for pollinators. So we kind of need that before we can do a lot of these other things in terms of proactively conserving them and improving our business practices and giving us the tools and information we need and communicating and collaborating internally and with our partners. So FLPMA section 201 also asks us to maintain on a continuing basis an inventory of all public lands and their resources and other values. This inventory shall be kept current so as to reflect changes in the condition and to identify new and emerging resource and other values. So again, another important passage for us to keep an inventory, but also kind of monitor those changes over time. And certainly, pollinators could be considered an emerging resource right now with the attention in our country pollinators are facing and the need for monitoring. You can see the map down there, though. One of the challenges we'll keep coming back to is how BLM land kind of sits in the matrix of other lands. And you'll see a lot of Alaska examples because that's where I'm at, but a lot of this is everything I'm sharing is pertinent nationally. But you can see a map here where the BLM land is in yellow, but we've got refuges in green, national parks in purple, state land in blue, tribal land in that kind of orangey color. And often BLM land is part of this matrix. We manage less large continuous blocks than some of our other agencies where they have these pretty discrete boundaries. So we're mixed in a lot. So how do we manage and how do we monitor those areas where it's not as contiguous of a block? It points to working with partners, but we can get to that later. We can do a lot with the information we have, but we do have a lot of information needs at the BLM for monitoring pollinators, and you can see a list of them here. This is kind of just a potential list, and the needs kind of vary locally as to what each office or what each state, what each planning area, what each habitat type or ecoregion might need. But as you know, there's a lot of different ways to look at monitoring and research for bees, and I think many of these will show up as needs for BLM across the land. So ultimately, we need decisions. We need information to support and inform our land use decisions. We're subject to doing NEPA analysis on everything where there's a nexus, either with our land, with our funding, or with our authorizations. Create that federal nexus. So we're reviewing the impacts and documenting those impacts, and where possible, avoiding and minimizing negative effects, and of course, promoting positive effects as much as we can. So we need this information to support and inform those land use decisions. When we're thinking about bees, we think about the management and conservation of bees on a few different kind of general levels. The first is sensitive species, and that's kind of in a broad sense, that term sensitive species. Of course, ESA listed species are going to be a priority. We have 17 BLM sensitive bees listed in 11 Western states. And I think that number will likely increase as those lists are revised, especially as we get more information. And then the state SGCNs associated with the state wildlife action plans list a number of bombus species, especially on those lists. And then generally NatureServe GNS rankings. And again, just all of these things seem to be changing very quickly, but we're looking to manage habitat and conserve these types of sensitive species. We're also looking to conserve and manage bee communities and their habitats as a whole. And then in the broadest sense, we're looking to preserve the ecological function that bees provide, especially the pollination function. Our assessment, inventory, and monitoring strategy has the objective to provide a standardized monitoring strategy to assess natural resource condition and trend on BLM public lands. Most people associate AIM with the specific AIM methodology, like the terrestrial AIM methodology, where we're out doing line point intercept, like you see on this picture to the right. That's AIM methodology for certain resources, but this is the AIM strategy that applies to all of BLM and all of the monitoring that we're doing. So as we think about doing B monitoring, we want to think about meeting the intent of this strategy. The AIM strategy provides quantitative data and tools to guide and justify policy actions, land uses, and adaptive management decisions. And here are the 5 principles of multi-scale monitoring programs that AIM lays out: structured implementation, standardized field measurements, appropriate sample design, data management and stewardship, and integration with remote sensing. So when we think about bee monitoring, we want to think about things in all of these principles and hopefully fit them well under the AIM strategy. Again, AIM does have a lot of specific methodology associated with certain resource monitoring. Here are some of our terrestrial AIM methods, which actually fit really well potentially with bee monitoring. We're going to be hearing from Patrick Alexander a little bit more on AIM and lessons learned from the terrestrial methodology of AIM, on how that was rolled out, and some lessons learned on a landscape scale for providing this national monitoring that AIM does. One of the things that we've built to add on to AIM, we call Apsi. It's an acronym within an acronym, the AIM, Pollinator Supplementary Indicator. And it does 2 things in association with the terrestrial AIM methodology. So with terrestrial AIM, you're out getting plant species richness and foliar cover and other measurements. And we're adding on flowering plant phenology and pollinator visitation. Although we're not identifying pollinators to species, we are counting visitation to the flowering plants on the site. We've done this for 6 field seasons now in 6 different states, and we have a lot of potential uses for this information. We're actually working with Pollinator Partnership right now to summarize the 6 field seasons we have and lay out some steps for the future for Apsi. But what I want to point out is that AIM, terrestrial AIM, Apsi, and bee monitoring all fit together in a very modular way that makes a lot of sense to build on these things. The value of all of these data collected at the same time, at the same place, is really high if you can coordinate it, which may or may not always be worth it, but it's certainly worth considering. So what we're here today to work on is establishing our BLM bee monitoring protocol. And we have this plan in place to develop this in the next couple years. And you can see the design here is the protocol designed in partnership with BLM, USGS, USDA, RCN, and others, so that we have comparable with data gathered by other methodologies, best meets the needs and priorities of BLM staff, is curated to allow integration with existing BLM databases, and results in the publication of technical notes and a protocol manual. And that'll all be done through field testing and implementation and expansion as time goes on. So a little visual here. I'm sure you all live this, but as we think about doing this on a BLM-wide scale, the different monitoring that's going on and piecing it all together is a challenge. We have a lot of monitoring that's already going on BLM land. Some of you are probably involved in that. And it's a challenge for the RCN as a whole. And that's why, I think that we think this fits well as an RCN kind of case study today. Is if we can figure out how to do this on 10% of the country, that's a really good start to figuring out how to do it nationally. So we might have a local monitoring project, and it might hit a couple different land managers. In Alaska, we have the Alaska Bee Atlas, which also covers, the whole state and multiple land managers. We have Department of Interior-wide coordination that we've just established with our DOI Pollinator Conservation Coordination Group charter. We have the RCN, which theoretically encompasses kind of all of these considerations, but then also we have these other kind of latitudinal and longitudinal groups working on Arctic-wide pollinator monitoring. We just had a workshop with the 8 Arctic countries in February. And then also things like the trinational coordination with Mexico and Canada, that's kind of cross-cutting things a different way. So, you know, this is the playing field, it just kind of from the Alaska point of view, and I know we could kind of build this over and over again for every state and all sorts of different scales. So it's just it's figuring out how to piece all these things together. We don't need to gather all of this data firsthand ourselves, but how do we get the information we need ultimately in the most efficient way? So what spatial units are we working in? How do we prioritize these areas? What are the metrics we're using to prioritize them? What is practical with the vast amount of BLM land that we have and only moderate funding? That funding could vary. We could have some high funding scenarios. We could have some low funding scenarios, but through the life of a monitoring plan, it's probably going to be in multiple different types of funding scenarios. And who actually does the work ultimately? And the answers probably vary for all of these, and I'm not posing any answers or asking you right now what the answers to these are, but these are kind of questions for the whole workshop today and beyond. In case you're interested in working with us, here are your contacts for each state. If you need any additional help finding people, I am glad to help point you in the right direction. So you can, I can get you emails for these people or find you more local contacts if necessary. So let me know, and feel free to get in touch after the workshop if you have any questions. But I'm actually going to pass this to Patrick Alexander now, who took this wonderful picture, by the way. And Patrick is going to share with us a few thoughts on the AIM methodology and how we can learn from that as kind of a case study of setting up a nationwide, a BLM-wide monitoring protocol and some of the things that we could do differently or lessons learned from that. So Patrick, are you ready?
Yep, I think so. And first, so for those of you who I haven't encountered before, I'm Patrick Alexander, so I'm usually the botanist for the Las Cruces District Office in southern New Mexico. But then at the moment I'm doing AIM terrestrial analysis for the NOC. So first, a little bit about using AIM data in the context of pollinators. So one of the core principles of AIM is that it's data that you're collecting once, then using many times. And so the whole idea of, you know, this isn't a pollinator specific monitoring program, but we hope it's useful for pollinators is kind of core to the whole AIM worldview. And my assumption is that some of the features that might be especially useful for in a pollinator context include plant community composition, which we have in terms of percent cover by species and just the total list of species on the plot, percent total foliar cover and percent bare ground, and also soil stability for all the ground nesting species out there. And so there's definitely some data that might be useful. There are also a few just things that you need to be aware of if you want to use AIM data downstream. First is you'd really have to kind of know what sample designs you're working with. Most of the AIM sample designs are spatially balanced, stratified, spatially balanced random sampling. The important thing for us in that is the stratified part. We're usually intentionally sampling different parts of the landscapes at different intensities. And so if you're wanting to go from the data on the plot to, you know, a number of acres or something like that, you have to put those sampling intensities, those weights, into the analysis to get a good output. So that's kind of one potential factor to worry about. And another thing that's also important to keep in mind with AIM is that when we have monitoring efforts that are more focused on a particular species, we'll often be out there, you know, at whatever the right time is for actually seeing those species. Whereas with AIM, we're usually dealing with a fixed sampling season, and we have a plot order that comes out of the sample design, and we try to do the plots in order. So that means that within the whole set of plots one might be interested in, there'll be some that will have been have data collected on them when it was bone dry and nothing was happening, and some where it was, nice and green and lush and all that. So trying to account for those kinds of factors would also be kind of an important thing to worry about. And next slide. Okay, now a little bit in terms of some of the lessons learned, both from things that AIM has done well and for some things that AIM has not done as well. This one is almost entirely things AIM has done well, so that's nice. So in terms of the methods, the general criteria we used to come up with the core methods for AIM is that we want to have methods that are going to be maximizing the trainability, how quickly and effectively you can get people up to speed, the accuracy of the data coming out of it, the efficiency in terms of the amount of effort it takes to collect the data, and utility across ecosystems. So terrestrial AIM is basically intended to be usable anywhere in the US in theory. It doesn't work quite as well in forestry ecosystems because we don't have as many of those, so we didn't build that in as much, but that's the idea. And so there are a variety of data of methods that we would have kind of liked to include just in terms of getting good data out of them. Plant production is one of the examples that tends to come up for a lot of the BLM. And we just kind of went through and tried to figure out, okay, which ones of these are really going to give us the best bang for our buck. And plant production data is awesome to have, but turns out to be at the high end in terms of the amount of training it takes to do it well, at the low end in terms of efficiency, and also at the low end in terms of accuracy, unless you're really good at it. So that's one that didn't make the cut. So that's kind of general things to think about in terms of choosing methods. If something AIM has done incredibly well is calibration. So AIM is moving away from kind of a world in which you might have somebody come into the office and be trained on field methods in their first year, and then basically be expected to just do them reliably and accurately for the next 20 or 30 years. And of course, in practice, we know that people end up drifting in different directions in terms of their actual accuracy. So with AIM, we do frequent calibration within the crews so that we have month-by-month data on how close together the measurements of different crew members on the same crew are. And so that then in the future, we can look back on that and have an idea of, okay, how reliable is this data? Were they collecting data that was at minimum consistent within this group of people? So that's something I think AIM has done really well, and we keep all that calibration data too, so that we can look back on it. One of the other things to keep track of in terms of choosing methods is just what kind of support the crews would need in doing these methods. So for instance, with terrestrial AIM, one of the big things crews usually need support with is plant ID, because these are often people coming in for a seasonal position from outside of state. They don't come in knowing the plant's here. And so it's important to try to have ideally staff in the local office who can help them with that. If not, make sure that there's a clear set of people that they can get in contact with and get support on all those parts of the protocols that require some amount of expertise that they might not have. The inverse of that is in doing the methods, you generally want to, as much as you can, reduce the need for that kind of support, reduce the need for specialized ecological knowledge that would be specific to a particular place. Now, next slide. Okay, and in terms of sample designs, so one of the things that I think has become steadily more important to us over time with AIM is a lot of the early thinking in terms of AIM sample designs was based on landscape level inference, trying to figure out what's going on in the landscape condition as a whole. And that's obviously valuable, but I think over time we've come to learn that, that having a little bit more of a focus on sub-question-based samples is also really helpful. And for instance, in a pollinator context, I would probably mostly be looking at, like, on an individual office level or a state level perhaps, what are the big things happening on the landscape that are going to give you constant analyses in your NEPA documents. And instead of trying to address those project by project, maybe put together a sample design that's going to give you kind of a general answer for at least your area as to, we've got a new permit application for drilling an oil well. What are the effects of those likely to be on pollinators? Because right now, a lot of the times, I think we would just have to say, well, there's probably an effect, but we don't really know what it is. But in any case, these are the two things you have to figure out how to balance: is how much of your effort goes towards just what's happening globally versus what are some of the potential impacts that we really need to have an understanding of. Also, timing fieldwork. So I mentioned that a little bit earlier. So yeah, this is kind of a messy thing in general. So with the AIM sample designs, we have this order that we try to do the plots in because that's how we get things to be spatially balanced across time, so that we're not doing all the plots in one area in June and all the plots in another area in July, and the data from the two areas looks different just because of that bias. If you're trying specifically to capture data on pollinators, that gets more complicated because, of course, you do want to be out there when the pollinators are there. So it would probably take some more thinking to figure out kind of what the ideal strategy is in that context. Also, something we didn't do as well earlier, a lot of our earlier sample designs tended to be really heavily stratified. There'd be a lot of strata. And over time, we moved steadily further away from that and trying to have the purpose of the stratification is just to have different parts of the landscape sampled at different intensities. So you just want to have, like, what's the minimum, like, 3 categories of sampling intensity, have those be your strata, and move on. Don't try to have them be all necessarily ecologically meaningful units, because what you're really capturing here is just the sampling intensity. So we've gone towards simplification on that end. Also, sample designs should be in databases. And this is something we've been catching up on and kind of having more of a backlog than we would like. For a while, we were getting sample design data from all the offices yearly, and they were kind of going into a pile. And eventually we realized, okay, we should have had some good databases built the first year as to where we're going to store this information, so that when we're trying to use the data, we know what those sampling intensities are and how to deal with them. Okay, and my last slide. So, and then in terms of QA/QC, overall, AIM has been a real step forward for us in terms of QA/QC. One of the things that's really important here is having centralized integrated data management, so that you're not having to, say, constantly do data calls out to offices wherever the data lives and get something back, but the data is in a central place where everybody has access to it. And you really want it to have as streamlined a data flow from the field to that central database as you can. Earlier on, we had kind of a model where we had local databases that would be at a field office and then get sent to the NOC at the end of the season. And that didn't work as well as we'd like. Because you've got some steps there of making sure the data gets transferred appropriately, and that can get hung up on one thing or another. And then in terms of QA/QC, you want to be sure you have explicit processes for how you're doing QA/QC in the field at the end of the season, and then also in terms of post-season if you find errors in the data. In terrestrial AIM, I think we've done really well on the first 2 of those, and we're catching up a little bit on the third one, because I think sometimes you have the idea that once it goes into the database, well, it's final. It's like, well, no, you're going to find errors in it sooner or later. I mean, they're there. You didn't catch all of them. You never did. You never do. And as I mentioned earlier, workflows for expert assistance in terms of both the protocols themselves, having lots of people around you can contact with to sort out any questions you might have about how to do the methods properly. And then in terms of, again, any specialized knowledge relative to specific ecosystems that might be needed. Also trying to really trim down field data forms that is only stuff they're going to be seeing in the field is very important. And we've mostly done a good job of that. We have a couple of kind of lingering cases where, for legacy reasons, we have something on the data sheet that you really can't answer in the field. And then that, of course, causes a little bit of confusion during trainings and so on and so forth. So just, you want to avoid that as much as possible. And last but not least, one of my personal favorite things to work on, build taxonomic databases early. I don't know exactly what the world looks like in terms of bee taxonomy, but in terms of the plants, basically we want to have some flexibility between states so that if you're in Colorado and you're using Acrofield's flora, you don't want to be constantly looking up, well, this is what Acrofield calls it. What is it in our database? You want that to be as seamless as you can. But you also want to have some centralized taxonomic data management so that you can basically translate everything you're getting in from the field into a common taxonomic framework for analyses where you want to do that. And we're kind of getting to that stage right now in the AIM world. So yeah, I would recommend starting that earlier rather than later, because there are a lot of names out there and it's kind of a pain to try to do all at once several years down the road. And I think that's all I've got to say. So thanks.
Thank you so much, Patrick. I really appreciate that. And I'm taking notes here on things that I can incorporate as we work through this. If anyone has questions for Casey or Patrick or anyone on here who's from the BLM about what they do, feel free to put it in the chat. We don't have time to answer questions for them right now. We're going to move right on, but they're eagerly waiting your questions in the chat. So reach out. I'm going to share my screen here. There, does that work? Does everyone see that? Long screen, let's try again, share. How's that?
Yep, looks good.
Okay, thank you. Thank you all for agreeing to attend this last minute sort of late notice mini workshop that we've put together. My name is Olivia Carril. I recognize a lot of you, you're all my bee heroes are here today. It's kind of a little intimidating to be presenting to all of you, but I'm also really excited. On the one hand, I think it feels a little like my dissertation meeting, but times 10, because you're all here giving me feedback at the same time. But on the other hand, it feels like a great opportunity to hear from all of the best minds as I move forward with designing something that's useful for the BLM. I have a few things we need to do before we get started here. So number one is make sure you have the chat up and ready for yourself so that you can write in any thoughts or questions you have as we're moving along. Go ahead and make sure you have that ready. Your fingers are ready to type. And then also in the background, pull up in your favorite internet browser, menti.com, menti.com, so that when we vote, it's easy for you to navigate there and do that. Menti.com, menti.com. Brianne, maybe you could put that in the chat just to help people if they're struggling. And then finally, if anyone thinks of something they want to tell me about later after today is over, I put my email address down there at the bottom of this slide. So feel free to reach out. I'd love to hear from anybody with any thoughts at all that you might have. Okay, so Casey gave us a pretty lovely overview of where the BLM's coming from in their incredible efforts to document bees on the lands that they administer. And I want to flip the coin over. So from my point of view, when I reached out to the BLM about doing this, my objectives for the BLM were, of course, to add to the inventory of bees that are known on BLM lands, tying them to plants and location and phenology. I also wanted to establish a baseline that the BLM can refer to sometime in the future when assessing the health of their bee populations. And I want them to be able to do this in the most efficient way possible, while also giving ownership to the BLM so they can do it independently of me or anybody else. And I want to be able to give the BLM, as Patrick and Casey both brought up, insights into bee population dynamics in relation to grazing or burning or natural gas and oil extraction or recreation or whatever other activities are happening on public lands across different districts and regions and all of that. And the idea, of course, is to do this in a way that allows the BLM to quantify their bee populations. So I'm thinking that with more solid numbers associated with bees, they can more accurately compare bees to components of the habitat, to soils, to forb composition. They can compare between different areas. So sagebrush in northern New Mexico versus sagebrush in southern Wyoming. And they can watch their bee populations change over time. Of course, all of this is happening with consideration of the fact that funding may change over time. So the protocol needs to be something that can flex or scale down or scale up as needed. The general idea is on BLMs throughout the West to find out which bees live where and in what sort of numbers. So these two characteristics, richness and abundance, are kind of the pillars of both inventory and monitoring. Inventory gives us more of a snapshot of who is in an area at the time of collection. You all know this, I don't really have to tell you this, but monitoring, on the other hand, is geared towards watching how this changes over time. And monitoring, it obviously relies on a solid inventory, but it also relies on quantitative numbers and consistency in how we choose to sample in our sampling method. So inventory is maybe more fun sometimes. It's more opportunistic. It avoids the areas where no bees would be because the point is to grow a list, whereas monitoring can be more measured with an effort put on recording the zeros as well as in documenting the ones. So what we're talking about today is by no means new or the only protocol for sampling bees, either on federal lands or even on BLM lands. And my goal isn't really to reinvent the wheel, but just kind of to refine it, to come up with something both specific enough to be useful for the BLM, but generic enough to be usable and comparable to things happening elsewhere. Think of it as like the SUV of sampling protocols. It draws from what's worked well in other methodologies and that would accomplish the BLM's goals of documenting their bee diversity and monitoring it over the long term. It's a huge goal. We're shooting for the stars here. It would be a good idea to have a discussion of whether monitoring is worth it, considering the known variability in healthy bee populations. But today we're going to proceed under the assumption that some sort of monitoring is better than no monitoring. And we're going to address multiple aspects of one sampling protocol rather than diving too deeply into one portion of a protocol. My idea is that this serves as sort of a springboard for future discussions, and I'm open to hearing from any or all of you after this is done if you have thoughts and ideas beyond what we get to today. So I'm thinking of the goal today is to sort of gather ideas and comments. So to start, we're going to take a quick poll. This is an interactive poll, so open up that menti.com that you had there and type in the code at the bottom here. It's 5015-8474. You'll see it in bright red at the bottom of my screen. And just, you know, limit yourself. Don't put like 20 things, but put in a few things. What are the most important components of a bee monitoring protocol from your point of view? And I'm going to stop sharing my screen so Brianne can share hers. Generally speaking here, I mean, obviously this is a big question and there are multiple components, but what things are the least variable? What most needs to be standardized and what most needs to be considered when designing a good protocol? This is just to kind of get our brains going here. And Brianne's going to show us. Nice, this is great. Flexibility within a framework, feasibility, replicability, and sustainability. Sustainability is a good one. That is something that's on my mind a lot, especially with the BLM. Bee richness and abundance with host plant associations within season phenology. I like that phrase. Repeatability is coming up several times in here. I see that a lot. Standardized method to capture the biodiversity. Consistency. I'm thinking too, and we'll talk about this, people having different levels of expertise and consistency. That different levels of expertise, I think, is something that really has to be thought about. Floral records. Multiple collecting efforts, passive versus active. Yeah, we're going to talk about these things today. This is great. Using protocols that answer your questions using a sampling method that best targets focus groups. I would love to hear more about focus groups. That's something that's tricky to me. Someone's bringing up databasing right at the top here. Labeled data needs to be thought about at the very beginning. It allows use of multiple data sources. Right, so overlaying on top of multiple things. This is great. And Hollis points out we'll definitely be touching on the idea of focal taxa in future workshops. Plants are key. Network analysis also ties to plants. Clear goals. Okay, great. So that can keep running in the background. Feel free to keep adding. Brienne, if you'll stop sharing your screen, I can move on here. But feel free to keep adding to that. It's all recorded and kept, so we can refer to those things again. Is my screen sharing okay? All good? No? Yes. Okay, good. Thanks. All right. Let me see if I can go forward a slide. Okay, so what I'm going to do now is give you, and I think many of you received sort of a draft of this, a really quick write-up of exactly what's on this slide, but I'll go over it. This is the skeleton of the protocol I'm thinking about implementing. I'd love feedback on all of the components. So what I'd like to do today is walk through each piece and then get feedback one piece at a time on what's worked well for you, what experience you have sampling bees, what's efficient, and what I'm missing. What am I missing from this? I'll walk through each piece, and I should point out that this protocol is based on one that's used by the USDA Bee Lab that they've implemented in a number of habitats throughout the West. So I set up a 1 hectare plot. It's about 10,000 square meters, so 100 by 100 that I sample once per month, or if time and resources allow, I'll sample biweekly. I tend to sample between the hours of 9 a.m. and 3 p.m. That seems to be the window where temperatures are conducive to finding lots of bees. And we should talk about this because Casey tells me he does it a little different in Alaska. And for each sampling event, I collect with my net on flowers for 45 minutes. During that 45 minutes, I try and make an effort to move entirely through the plot. So I walk around, I try and spend about 10 minutes per quarter with 5 extra minutes for any areas that I missed. I do that 45-minute sample twice on a given day, and I sample with another collector who's collecting at the same time that I am, so that it's a total of about 3 hours of collecting in a plot. In addition to net collecting, I also put out a series of 30 pan traps. I've been using alternating blue and yellow. I know that white is also something people have been using. And I have had habitats, particularly like canyon bottoms or riparian areas, where a 100-meter square plot was not representative of a habitat. It kind of went over a couple. So in those cases, I've adjusted the plot to be 50 by 200. So it's the same area, but a slightly different shape. So we're going to walk through each of the pieces of this a little bit at a time. And I should point out that at the same time, and I'm showing a really old data sheet here just because it gets the point across, I collect data about the plot. And what I generally collect is the time of the collection event, the date, the identity of the 2 people that are collecting. And then I also collect information on where this is happening. With the plots, usually we know the UTM, so those don't get written in. I record information about average weather, evidence of grazing on a sort of a qualitative scale of 1 through 5. In this particular study, that was an important thing to consider. And then I record all of the plants in bloom in the plot. And I've used a log scale for quantifying these. So it's how many are there. If it's between 1 and 10, count how many exactly it is, round to the nearest 10, round to the nearest 100, which just gives a sense of relative abundance and of presence absence. And I don't know if you guys can see through the slides. Someone drew a picture on the field notes on the back, so I threw it in just for fun. This is how to catch bees on Tamarick. So there you go, in case you were curious. So you can see in my technique, I'm using 2 different collecting techniques, and based on my experience and what I've read, they seem to complement each other well. There are other ways to collect bees too, which have their own advantages. Net is great because it gives you a record of what the bees are visiting. The downside is that it requires practice to do it well, and it only works while you're out wandering around, so it's active collecting. Pan trapping requires much less experience. My 6- and 8-year-olds do my pan trapping for me in the summers, but it has been shown to oversample some bee groups and undersample others. And more evidence is suggesting that it collects fewer bees when there are a lot of plants out and more bees when there are fewer flowers. So something to think about. Malaise traps appear to be pretty successful in some environments for catching certain groups of bees, same with vane. A few studies have suggested that they're good at sampling a wider community than just what's in that 1 hectare plot. So they're less tied to the floral richness of the place that you are and more tied to the larger habitat. But they also tend to oversample, and there can be considerable redundancy in species. Malaise traps are nice because they can be left out for weeks. But the other part that's kind of hard about them is it takes a bit of time to set them up and take them down and maintain them. And there's considerable bycatch of other insect groups beyond just the bees. Obviously, each of these groups, each of these techniques requires standardization, what colors to use, how long to leave it out, how big should your pan trap be, should it have little nectar guides in there, how many to use, how high off the ground to put it, etc. But we're just going to talk briefly about this. I'd be curious any thoughts you have. I know we don't have time for everything that everyone wants to say, so feel free to comment in the chat. Remember, we're recording all of this. All of this is good information. And I should point out too that you're going to have time to talk in your breakout groups. When we take a break here, when we come back, you'll be able to share. So don't feel too bad if you don't get a moment to talk now. But if there's anyone who has an idea right off the top and wants to raise their hand or speak up in some way, I'd be happy to take any initial thoughts on sampling technique. Don't all speak at once. Jason, go ahead.
I mean, I've used, I think all of these at some point, and my experience with blue veins is that they tend to really overrepresent uterines, sometimes agapostamins and some holyptids, and a lot of obviously bumblebee queens and things like that. So although I use them more for inventorying, and I'm happy with your decision not to use them for the monitoring.
Okay. Thank you. I appreciate that. I really do. I was on the fence. I've actually been thinking more like maybe I should be including them because I'm missing something otherwise. Lucia, what do you have to say?
Hello, I'm Lesha Tronstead, and I've been doing a lot of bee work for the BLM in Wyoming. And we've done a lot of comparative work with pan traps, vein traps, and netting. And I have to say, I'm a big advocate for vein traps, but I also have to say that they have to be used carefully and correctly. I see a lot of studies that leave them out for an extended period of time, and I don't think that's a responsible way to use them. We put them out for a maximum of 48 hours, and we don't put them out until queens are in the ground, queen bumblebees. But we find they collect a wide range of bees. They collected 97% of all the taxa we collected using those 3 methods, whereas the pan traps only collected about 75% of the species. So we find that they do a better job of collecting the bee community in general. Yeah, they did a great job, but they have to be used carefully. Oh, the other thing I wanted to say about veins is that they do eliminate processing time in the lab because there's no rain. Then you don't have to wash the bees. We just keep the samples in the freezer and then pin them, and you don't have to wash or dry them. So it's cheaper.
That's really good. That's really good. Will you put some of that in the chat or in our breakout room so that I can remember that? Those are great comments. Matt Carlson, go ahead. I'm sorry, I accidentally lowered your hand.
That's fine. That's fine, Olivia. Yeah, I was curious about how you decide on where these plots are located. What's the relationship with the BLM and habitats and so forth?
It's a great question, and it's one we're still figuring out. So the BLM, I'm hoping, will give me some insights into things that are interesting or concerning or exciting for them, and that will help guide me to the location. And then on the ground, it'll be, well, we'll talk about that a little bit, but I really think it's going to be a little bit of intuition about what makes the most sense. Considering funding and how long it takes to get enough data to really say something, I want to make sure that my few plots that I'm putting in end up in places that give me a lot of bang for the buck, if you will. So it won't be random. I don't think it makes sense to do a random plot, but the general area where it happens will be decided, I think, by the BLM. It'll probably be a conversation between the two of us.
Olivia, You've got some, just want to make sure you can see them. I know while you're screen sharing, it's hard to see, but there's some stuff in the chat too. Just FYI.
Awesome. Okay. I have time for one more comment. We haven't even got to sampling unit yet, which seems like a big one. So let's take one more comment and then move on to talk about the next thing. Knowing, remember, there will be time later. So Terry, I saw your hand first. I'll go there. And Eleanor, if you can save that comment or put it in the chat, I'd love to hear your thoughts on that a little later on.
Well, just having worked in some projects where they use both vein traps and pan traps, I think one of the things to think about in terms of their efficacy is the ratio of pan traps to vein traps that you have, because it's going to make a huge difference, I think, in your coverage.
Okay. I
And
hope you
I
end
think
up in
also
the same breakout room as Lush. I'd like you two to compare notes on that. I'm curious too. Thanks, Terry. That's great.
Yeah.
Okay, I'm going to lower your hand and we'll save those comments. Eleanor Lichtenberg will come back to whatever you had in mind there in just a minute. I think I'm still sharing my screen. Is that right? Am I doing okay there? All right, then I'm going to move on to my next slide. We're going to talk about another little Menti question here. So how many people should be participating in a sampling event? I've always gone with 2 because I find that we catch different things and it seems to make for a more complete sample, but 2 might be hard if funding is tight. And if you're just putting out pan traps, you know, 1's obviously sufficient. So it might depend on where the sampling is happening, on what kind of sampling, maybe 3 or more for a short duration, like an intense, I don't know, a whole bunch. Just something that crossed my mind. All of this, of course, considering the costs and time of multiple people. How many collectors, just a vote on Mentimeter there, would you say are adequate when sampling bees with a net? Again, any extra comments, we're not going to comment on this specifically right now. Put your thoughts in the chat or save them for our breakout room. There will be time for that. This is just a quick vote to see, and I'll stop sharing my screen so Brianne, you can quickly share and show what's coming up. And then we'll come back to that again at the end. Two, lots of 2s. More than 3. Whoever put safety is a concern. I know this is on my mind as well, especially some of the remote areas where we end up in. I always feel more comfortable having 2. It occurred to me that 3 might be a nice option if one person was in charge of like plot monitoring and the whole goal was to accurately record the flowers, other stuff about it. It might be interesting to have 3 that did that. Strongly agree for, so yeah, that's good. I see one person put more than 3. If you wouldn't mind putting in the chat why you think more than 3, I'd love to hear somebody's ideas on that. That's really neat. Okay, so I'm going to now, let's see if I can do this. I'm going to share my screen again. And we'll move on here. I want to talk now about a really big one. This is sampling unit. And this is a pretty big topic with a lot to consider. In terms of inventory, I kind of wonder how representative a 1 hectare square is of a much larger habitat. And as for monitoring, if you only have 1 plot in a big area, what if you pick the 1 spot where very little's happening and it doesn't end up representing what's really going on that landscape? Especially considering that putting out multiple plots is a lot more time and a lot more person power. So what's the balance between those and how do you decide? I'm going to bring up some data to demonstrate one of the reasons that this is on my mind. And this is from a study I did a long time ago in 2001 in a large national monument in southern Utah. And this particular year, 2001, I had about 40 plots that I sampled, and I sampled them every 2 weeks for a 6-month flowering season. So this is showing the similarity of each plot to each of the others for that year. So plot A to B, C, D, E, etc. Plot B to all the others, and on and on. And it's based on all the species collected across that 6-month period. I used a Bray-Curtis index here, so it includes a measure of abundance. And what you can see is that plots that are closer together in space are definitely more similar to each other, but that by about 6 miles apart, plots become pretty dissimilar. Interestingly, plots that are in the same habitat or at the same elevation tend to clump together even over longer distances. So you can find that bees in high elevation communities tend to be more similar over longer distances. Interestingly, the similarity between plots can be attributed to just a handful of species. I found in this particular year, 38 bees were found in more than a quarter of all of the plots. So we caught around 350 bees that particular year in plots, and about 200 of them were found in less than 10% of all of the plots. So what this means is that a handful of the bees provide the majority of the similarity between plots, while many less widespread bees are providing what makes them distinct from each other. So in my mind, this makes me think about which is more important when monitoring: changes in the less common bees or changes in the common bees. The less common ones maybe are more sensitive, or maybe they're tied to a particular habitat, but the more common ones are easier to get large amounts of data for. I mean, maybe one plot is enough too. If it's sampled against itself over multiple years, perhaps a thorough sample in one place is enough to say something about bee populations, but I do wonder, what if that plot just happens to be in an area with few bees or poor plants? If few samples are to be made, then it makes sense to me that you might pick that place very carefully based on your knowledge of bees so that you don't end up missing the majority of the data. I want to point out too that there may be other ways to sample bees besides my 1 hectare plot idea. What about some variation of a modified Whittaker plot like they use for plants, where small subunits are pulled together to create a more complete picture of an area? Practically speaking, this might be very expensive. However, it could give an idea of a species area curve, maybe after all the bees are identified, so likely not in the field. I don't know. Or perhaps rather than focusing on an area, we should focus on a plant. We could sample rabbit brush for 10 minutes at a time and do this all across the West. And you could pick one plant that's ubiquitous early in the season, say Opuntia prickly pear, something like that. Another in the summer, maybe helianthus or sunflower, and then a fall plant like rabbit brush, Ericameria. And you could lump multiple samples together in all sorts of various ways and look at changes between years. Or, and this is another interesting option, what if you just sampled 50 bees and see how many species you get in different areas after having 50 total specimens? Or maybe how many minutes it takes to get those 50, or how far you had to walk to collect them, or how many plants it took to collect them. I'm not sure about this. It's been brought up to me by a couple different people, so I'm throwing it in here. But I have to say, there have been days when I've been out in the field at a site and I would never have gotten to 50 bees. You'd have to wait till the next flowering season to pull that off. So it's hard to say. So let's discuss. I think we have time. Let's see what we're at. We have time for a little over a 5-minute discussion here. Again, put those comments in the chat. I might not see them all right now, but I will see them eventually. Does anyone that hasn't spoken up want to say anything? Eleanor, I think, is your hand still up about this one? Okay, go ahead.
Yeah, so this is sort of more a general comment rather than a specific question. So I'm actually in Texas, where we don't really have, I think there's a tiny bit of BLM land, but I'm working in rangelands and grasslands. So I think there's a lot of similarity with BLM lands. And one thing that I've sort of been thinking about has been, and that differs from a lot of the published research in crops, is that out in grasslands and rangeland, sometimes you don't have that many flowers, or they're super dispersed. It's super heterogeneous, and so I'd be interested to hear what you think. Maybe we don't actually have time to get into this, but this issue of grasslands being very different from sort of crop agricultural landscapes, and how do we account for that? And like you said, yeah, some days there's just not much out there, and it would take a week to get 50 bees maybe.
Right.
And at least in my case, I'm also working around, students' class schedules, and also things like it being too windy. I presume a lot of BLM land also has this issue with just these large open spaces. We have some, so just I'm interested to hear people's thoughts on dealing with that.
Yeah, I mean, it's a big deal to come up with something that's standardized, considering the variability, not just of the bees across a season or between years or whatever, but variability in the human component also varies and can be tricky, and the landscape varies. Yeah, so what's the common thread that can tie all that together? Yep, that is the question of the day. Any question? Patrick, go ahead. You have a question or thought?
I think it just disappeared from my head. Uh-oh.
When it comes back.
Oh yeah, sorry.
You got it? Okay.
Yes. I was gonna say one thing we do in some of the AIM methods is have the time that a method takes be flexible based on the complexity of the plot.
Okay.
But have an explicit stop rule. So
nice.
for species richness, it'll be something like you go for at least 15 minutes and you keep going until either you've covered the entire plot or it's been at least 2 minutes since the time you saw a new plant to stick on the list. So that means on simple plots, it might be 15 minutes. On complicated plots, it might be an hour, you know.
Right.
So having that might help deal with the fact that sometimes you're out there and there just aren't bees and you don't want to just put a ton of effort into that. So
that's great, Patrick. Thank you. Ty, go ahead.
Yeah, just one comment on the maybe the 50B method. So I can sort of imagine it as a cutoff when it was really, really hard to find any bees. But in other plots, you would really have to sample randomly around, because you can have one plant with 50 bees on it, and then the bias of where a collector goes would overwhelm everything else.
Yeah, no, it's not necessarily the most practical in my mind either, but I thought it worth bringing up just because it's been mentioned to me so many times as something, and I thought maybe I'd missed something. Yeah. Other thoughts real quick on sampling unit? Anything you want to say that might spur our breakout groups specifically? Lesha, go ahead.
I just want to mention that I did a power analysis recently, comparing the 3 different methods again, vein, bowls, and netting. And so what we have done is net for 30 minutes actively. And with the netting, you need a lot more samples to detect a change, because you have that set time, or I think the same thing would happen with a set number of bees. So I think that makes it really tough to detect changes in abundance, but it does really help with the richness trends over time. So,
and it gives you that floral component that you wouldn't get any other way.
Well, we have started doing quads floral surveys whenever we're out there doing bee work, because it is so important for passive traps to know. We count blooms per species, so how dense are the flowers, and surface area of blooms has come out as the most important for us.
So cover, percent cover?
We just calculate the area of each flower bloom.
Got it.
And so, like, some plants put out one big bloom, whereas like phlox puts out a bunch of little ones.
Right,
so,
right.
Yeah, we've done flower area as one metric,
okay.
And that came out as the most explanatory one. But yeah, anyway, we always collect flower data now when we put out passive traps, because it explains a lot of variation of what's going on. And I think using a complement of active and passive is really important, because like you said, some areas don't have many flowers, so the passive traps will work better, whereas some areas, meadows of flowers, the active works really well and the passive isn't as well. So I think using both kind of covers you in those types of situations.
Right. Yeah, that makes sense. Thank you. Okay, save any more sampling unit. How best to sample, whether 1 hectare is good, whether it should be bigger, how many in an area, how to figure that out even. Thoughts on that, I'd love to hear. I'm gonna move on a little bit here and talk about how long a sample should be, which gets at something Lesha was just bringing up. This is a new Menti, so you're still in menti.com, but notice that the code at the bottom has changed. So enter in this code. I'll stop sharing my screen so we can watch this happen live here. How long in a plot? Now, this is over the course of a whole day. Is 60 minutes, 60 total minutes, maybe split between two people, so 30 minutes each. Is that enough? Do you think, based on your experience in the field collecting bees, is 30 minutes across two people or 60 minutes with one person enough to really say? Maybe some of you have analyzed your data and actually can speak to this in a way other than just your general impressions, but I'd be curious any thoughts you have. 90 minutes maybe is better, 120 total minutes across a day, or 180. 180 is about 3 hours. That's what I have done in the past, is a total of three hours split between two people, but I'm curious. Any other experiences with how long to sample in a plot? More sites. Jason says he'd pick more sites over more minutes. So maybe a shorter amount in a place, but across more places on a day. Sure, I can see the value of that, actually. It seems like you'd need at least 3 sites per area per stand to even start at floral or bee diversity. Yeah, I can't argue with that. These are great comments. I can't wait to read them all. Okay, I'm seeing, so far we have 22 votes up there, I'm seeing. And it looks like in general, a total of 60 minutes spent in a plot on a given day is maybe sufficient in most people's eyes. On our protocol, we currently do a total of 45 minutes collected and usually spend about an hour total minus photos. So we were able to get 3 to 4 plots a day using this. Driving was not an issue. Cool, that's great. Okay, that will keep running in the background. If some of you are still taking a moment, I'm going to go back and share my screen. I have a few more of these for you. I hope you don't mind. It's a great way, I think, to collect some data here. I want to bring up a thought that I had on this. It seems that one way to standardize between studies that have sampled for different periods of time would be to find sort of a common denominator. So a small unit of time that could be lumped together to make bigger units that could be compared across disparate studies. So if someone's sampling for 30 minutes, I'm sampling for 45, but I had it broken into 50-minute units, I could take 2 of those. I could take any 2 of those, randomly even, and pull them together to make something comparable and compare. This would, of course, make for some interesting species accumulation curves across a day, but it would require more manipulation of samples in the field, which maybe is a drawback. So a thought, if you have any comments on that, put those in the chat. So my next question is, does it make sense, and I know someone brought this up in the chat, to sample a plot more than one time for a day? If you're sampling at 11 o'clock in the morning, is that the same as sampling at 2 o'clock in the afternoon? And if you are sampling for, say, a total of 60 minutes, does it make sense to break that up across a couple days? So again, I'll stop sharing so we can see as we're going here. Jason, I'd love to hear the practicalities of how you did that with like vials of different flowers and stuff. It seems like a lot of different collecting tubes or something to do that. Twice seems to be so far what most people think, because you do get different, good students, great answer. You do get different bees at different times of day. That does seem to be a thought that I have as well. I do find that by starting at 9, if there's any like really early in the morning bees that are kind of done by the time I get out there, I might miss out on those bees. Hollis says she's the more often person. So Hollis, when we have a minute to chat, I'd love to hear your thoughts on why more often than just the twice. I'm going to guess maybe it has something to do with what I was just saying. That's good. 20-person minutes. There's a lot of things in the chat too. Hopefully we can come back to some of those. We're going to have more time at the end. I want to make sure I get through all the slides. Okay, Brianne, I'm going to go back here. And then we'll hopefully have some extra time here and we can talk through some of these. Okay, so now moving on a little bit here. As far as pan traps, what are your experiences when it comes to how long to leave these things out? Obviously, we talked about blue vein traps being left for just maybe 48 hours, I think, maybe not any longer. I know it depends on the kind of trap you use. That might not make as much sense for pan traps to leave out for 48. Since I'm using pan traps for the most of what I'm doing so far, I'm going to limit this answer to pan traps just because it will be more useful to me. I'm being a little selfish on this one. But if you have thoughts on those other traps, I'd love to hear your comments in the breakout rooms. So again, notice that one more time, this is the last new Menti code that you'll use, but there is a new code there. What do you think when you're dealing with pan traps specifically? We're talking about pan traps. Is 4 hours enough? I know that in the Fish and Wildlife, they talk about maybe leaving them out for a total of 24 hours. Is there value in that? Or does it just mean you end up with so many lasioglossum that it drowns out everything else and oversamples? I'm seeing lots of 24 hours, 6 hours, one for 12 hours, 12 hours. Interesting. I'm in the 6 hour block. That's what I've done in the past. But I think what I'm going to do this summer is maybe put pan traps up, maybe in my backyard here. I live on 4 acres or so. Put them up for different periods of time and just see at what point I stop getting new bees in my traps. It might depend on the time of day. It might depend on the time of year, not time of day. Logistically, definitely, Terry, it is a problem because you have to revisit that site and that can be tricky. Especially with, you know, if it's underfunded staff. Here's an interesting comment. Let's see here. If this method is adopted by BLM field office teams, it would be for a set amount of time, like the time of running an AIM plot for 1 to 2 hours. Interesting. Returning to sites to pick up trays causes reduction of plot work. Right. We would need to be focusing on independent team for that sort of work. Yeah, that makes sense. AIM plots definitely are for shorter periods. And I have a lot of AIM samples here because they've put pan traps out on my sites in New Mexico. And I know they're out for just a very short window. We've still managed to catch some interesting bees, but it doesn't feel like it fully represents what might be in a sample. Lesha says she finds her pan traps usually evaporate in 48 hours in sagebrush. So that's also interesting. Okay, Brienne, we're almost through these votes here. I'll share my screen over yours. Almost done with these here. All right. So with all of that, any overall thoughts on how long to sample that should be brought up here, sort of to spur our discussion later in the breakout groups? Anything that I'm missing that we need to be considering? Anything from your chat that you feel everyone needs to really hear before we split up into different groups to talk about this? It's a lot. How long to do this is a big question with a lot of nuance to it because it depends on technique. It depends on all of that. Casey, go ahead.
Yeah, I put this in the chat, Olivia, but just a reminder for everyone, and I don't think I think everybody knows this, but it's such a challenge to think about the scale, you know, and I think every answer to all of these questions needs to be considered with the huge scale, you know, 240 million acres or so that we're operating under to get this done. Now, it doesn't all have to be done in 1 year. Maybe if we can plan it out for 10 years or something, that's reasonable. But, you know, we need to balance all that with the value of the information we're getting and the amount of detail that we're getting from each plot. So it's a tough balance to find that sweet spot to be, you've traveled all the way there to this point to get good information at this spot. You know, how much time is it worth spending there until you go to the next spot and have to keep going and going and going for 10 years theoretically, conceptually. So anyway, just a reminder for the challenges of all this.
Yeah, no, absolutely. And I think, you know, in my mind, sort of the way this would roll out and that we've been talking about is that we would try something and use whatever we learned from that to slowly over time find that sweet spot. I think the sweet spot is definitely going to be a trial and error to really get to. But yeah, 240 million acres is a lot. Wendy, go ahead.
trying to get my mute off. So yeah, I agree with Casey, and that is our biggest problem. You've got the scale of drive time too, because in a lot of my areas we're really small scattered parcels. So you've got 2-3 hours in between drive time and then setup and all that. So I was going to say, yes, we've used the propylene glycol also in some of the grassland sites if we've had to leave them overnight, because they will evaporate if you just use soap water. And so one thought that I'm having is, you know, BLM, all of us pollinator people probably should have like the internal discussion also about what level do we want your study, Olivia, to cover for us? Because I can see if you can spend more focused time to get that broader diversity question answered for us, might be the best use of your time and our money in that sense. But then being able to figure out how do you neck that down to make it a usable thing that we could add to field office staff, if that's something that we're looking at. One of the reasons we're not using the Apsi, the AIM supplemental protocol here in Montana, is it takes too much time. And, you know, we're focused on getting our AIM plots out there. And so adding pollinators, that's why we ended up developing the photo modeling protocols to try to figure out if that would work. And I will say preliminary data is saying that we're between like 89 and 91% accurate on photo ID. If the photos are taken by a good macro lens and we're getting all of the right photos that we need for the identification. But the problem that we do run into is there's so many different morphs in the males for some of our species that they get a little more challenging for some of the species. So anyway.
I'm curious, are you able to photo pretty much everything? Are there certain groups that are just too hard to get a good photo of?
Well, so we're working bumblebees only right now.
Okay.
That's the big difference.
Yeah.
I don't know how. Yeah, I don't know how that works with things like megaloides, because they're
Neither do I.
you know, exactly. So, but we're trying to figure out how do we take this photo protocol and move it up to all pollinators? Because you get into things like skippers. We've got Dakota skipper, which is a T&E species, but there's tons of other skippers that look very, very similar. And so having the expertise that really help us focus in what are those critical ID markers that you know, taxonomically you would need could help us. But then also in my mind, we've got to figure out what is our broad scale diversity of all pollinators, because we talk pollinators and there's so many different requirements across the different groups. So that's my other big headache in this is, you know, it's great that we can focus on something like bumblebees or bees in general, but there's much, much bigger pictures out there too. And we've got to start somewhere. So that's why it's if whatever we develop, if we can move it into more of a broad scale process, that's going to be the most beneficial over time in my mind, if BLM takes this on as our own responsibility at the field office level. If we're going to keep it more at the national level, like what we're doing with you, then we can ask different questions.
So that's great. And I'm curious now, I know there's several BLM folks that are on here today. I don't know, I'm going to guess most of you are at the state level, just looking through names that I see on here. If that's the case, this is a weird question, but if you wouldn't mind putting in the chat, on average, how far of a drive is it for you to get from your district office where you park your personal vehicle every day out to a field site? Is it generally that districts are, you know, 30, 40, 60 miles away from any field site at all, from any location, or are you more located close? I'd love any thoughts from the BLM on how long it takes to get to the field for you guys.
You're getting some answers in the chat, and it's a long time.
It is?
Yeah.
Okay.
1 to 3 hours, 3 hours, and 1 to 4 hours are the answers.
Okay. So it's a full day trip just to get to the place where you're going to do a plot. That's really interesting. Okay, I think we're doing great. My goal was to be ready for us to take a break and stretch before our breakout groups by, what do I have here? By my time, 12:40. It's 12:25 right now. So I'm going to ask you one more Menti question, a last thing to vote on that we haven't really considered. And then I'm going to throw out 3 really big picture questions that I'm hoping we can talk about in our breakout groups. And then if we still have time, which I think we will, I'd love any comments that we have right up until break time. So I'm going to move on here since I'm still sharing. The one thing we haven't talked about, and this might matter to BLM folks that are driving a long way, is how often do I have to do this? If I'm responsible for taking care of some plots in the middle of nowhere, how often do I have to go? So with the constraints that are obviously clear for BLM folks and distance to sites and all of that, what's the minimum that a site could be sampled? Would it be okay to do it once a year, but always do it, you know, in May or whenever peak flowering is for your particular area? Maybe it's June or July in Alaska. Or is that not enough to really be able to say anything meaningful about an area? So the Menti code is up there for this one. I think it's the same as the last one. I can't remember, but I'm going to stop sharing so we can see each other's answers on Brianne's screen as they come in. Bi-weekly, the hard one. Yeah, if you sample multiple times, you'd be better able to compare across years. It's true. Phenology will be different from year to year, and I've seen that plenty of times in my own work as well. Wendy says seasonally because floral resources over time. Yeah, so Diane brings up that you could focus it instead of focusing on all the flowers, you know, find the flowers that are most interesting, either because they attract the most bees or maybe because they attract species of concern, and put the focus on when those are at peak bloom. And pair up sites. So here's an interesting idea. This is from Eleanor again. She ran a study with traps for 24 hours where each site was within approximately an hour of where she worked. Now she can leave traps out for 24 hours and pair up sites. So 2 sites in 2 days. Because floral resources change. Thanks, Wendy. Okay. Pretty animate. I've been doing 3 weeks here in New Mexico too, and I've been pretty happy with what I've found. You can see I didn't put that as an option here. Maybe I should have, but I'm glad you brought that up. 3 weeks is good. Terry suggests, and this might be worth discussing, in many places there are kind of 2 flowering seasons, especially in the West. So maybe you could go with one in the spring and one in the fall, or just one in the spring when most of the flowers are out, or vice versa. Okay, great. So let me share with you 3 sort of big picture questions in case anyone has any opinions on them and would like to share their opinions on these general ideas. I'm open to hearing everything. So there's 3 things that kind of are important to me that I'd love to hear any thoughts that you have, because I'm open to trying to fit something like this in. So the 3 questions, and these are related to each other. If you could answer any question about bees across BLM lands using them as your giant laboratory, what question does it seem to you is most important to try and answer about bees in the near future? I'm not talking about lab work. I'm not talking little behavioral studies, more general big picture questions. What might we try to answer with this? You can put something in the chat, but I'm going to tell you now that in the breakout room, you're going to have time to sort of type in or speak your answers to this. So just kind of think about it in your mind right now if you'd like to save your thoughts. And along those same lines, is there something we don't understand about sampling protocol that it'd be great to figure out for all of us in this next year? With me getting ready to implement this, I'd be happy to try and tackle or get data for one of these sampling protocol questions if there's something we don't have a good enough answer on. And then finally, and this is bigger, and maybe we can take 10 minutes now to talk about this or anything else you want to before our little breakout. How would you each of you, structure a rollout of sampling protocol across the BLM? Would you start with one state and then go out from there? My first thought when I sort of approached the BLM about this was to do it in sort of pick one longitude and do it as a latitudinal gradient going up and compare all areas from New Mexico to I guess Montana, and then the next year do Arizona through Idaho, and then et cetera, and move on. But maybe there's a different way to structure this that might be informative and useful. I want to say too, and it's important, I think, to make sure that this is brought up, that I do know that this is a whole mini workshop in and of itself, talking about what to do with the data after it's been collected. So the BLM and I are discussing very much, and Patrick, you mentioned this in your talk as well, the labeling, the databasing, the identifying and curating of whatever samples are collected over the long term. So I just want to acknowledge that that is a big topic that's also happening, but I wanted to focus on the sampling protocol today. So next up, we're going to take a break, but we're running a little early, which is great. I'd love to hear any thoughts you have on anything we've talked about so far. This is your chance to pipe up with some thoughts before we break, and then formalize all this in our breakout rooms. If you were me, would anyone who likes the idea of south to north sampling in 1 year? I thought with climate change, if that were repeated every couple of years, that might be interesting in one habitat. I see someone else. Eleanor had the same question.
Seems like people like that idea, just looking at the chat.
Okay. Attitudinal sampling. Yeah, you could pick just one habitat. I don't know, something. Pinyon juniper is fairly common. Sagebrush at higher elevations. You miss the hot deserts when you do something like that. Or maybe it's something about the composition. Maybe wherever the area that shrubs compose of a landscape is a certain amount, you sample there. Maybe all woodlands. PJ doesn't have many bees in my experience either, Terry. Not much bang for your buck there. Grazing impacts on pollinators is on my mind a lot. Yeah. Patrick suggests we could change our sampling to include all of the little biting insects in the PJ there. One thing that comes to mind from reading these comments is, do we need separate Western and Eastern protocols? If we did, Eleanor, where would you fit? Are you Eastern or Western there in Texas? Might be hard.
Both. And also North and South. But
ideally, whatever we come up with would work anywhere, right?
Yeah, so I think having lived and worked in different parts of the country, a big thing I see is a difference between grassland versus areas that have a lot more trees.
Okay.
And I think, I was running a grassland bees journal club for a little bit, and one of the things we pointed out was that a lot of the past sort of more theoretical work was done on the East Coast, where forest was the native habitat as opposed to our more open habitats.
Hollis, go ahead.
Well, this is less about sampling per se, but one thing that you might consider is some sort of analysis of where BLM personnel are in relation to BLM lands, and get a sense of who's like willing and able to go out and collect traps or net, like collect data for you and or with you. So
right.
That seems like it would be, I think the people getting an assessment of the people power and some of the logistics of travel would be really helpful, because I could see that being a limiting factor.
What about where to prioritize within? I mean, based on our understanding of bees and what areas haven't been sampled, what areas of the West most need to be sampled? Is it our pristine landscapes? Is it the national monuments that are run by the BLM and the, what are they called, NLCAs? Or is it Did I get that acronym right? I think so, something like that. Or is it instead the areas that are more heavily disturbed, where grazing is really intense, or where I don't know, they're thinking to treat for sagebrush or some of these other things that happen on BLM lands? Which is the priority? And here's another thought I'm just going to throw out. I don't know how many of you, I think most of you are probably aware of what Andoni and Link have pulled off in Oregon with all of those volunteers out there opportunistically collecting bees in their souped up bee vans. The volunteers that they have there and what's going on is pretty incredible. And it's crossed our minds that perhaps volunteers would be willing to help the BLM by doing this monitoring once the protocol is up and running on lands for them. Does anyone think that would work, or would it be too disappointing because it's not opportunistic and there's times when nothing's happening during monitoring of a plot? Nevada is undersampled. So this is the fun part, is that a lot of the people who volunteer are retired and willing to travel, and they don't mind not being near a site. They're willing to head out and take a little adventure. Casey points out that a scalable protocol is important to allow for different habitats and funding and the potential to incorporate other bee sampling efforts. For sure. I'm seeing several Great Basin, and LCS. Yeah, what is pristine? It's true. It's moving scale. Yeah, good. These are all great comments. Is rabbit brush common from north to south? It's certainly common where I am. It's common in Oregon and Washington. It's common in California and at least northern Arizona, but I'm not sure about southern Arizona. Okay, I'm seeing great comments in the chat. I can't wait to read through everything. I say we take a 13-minute break. Meet back here in 13 minutes. We'll divide you up into breakout rooms where there's plenty of time for everyone to say whatever they want to. Hello, welcome back. I hope you all had some interesting comments. I can't wait to hear them. I wonder if one person from each of the, I think it was 5 breakout groups that we had, if one person from each would be willing to discuss what they found in there. And to structure this, Brianne, can you, I think you had the groups numbered, is that right? Looks
yes,
like
they were numbered.
Okay, so you shot out a group in that, well, I guess I don't know my group number. What was I? I'm group 1, I got it. So I'll go first. And then group 2, you guys can go after, etc. So we got a lot of comments on the quiet writing question part, adding comments about each of the components of the monitoring program. I'm not going to read through all of them. There was a general consensus that less time could be spent in a plot if it meant that more plots could be hit in a certain day. We also had someone point out that maybe trap nests should be part of this. They could be included in it, or that if you did a blue vein trap, you could just put one in the very center of a plot and that might suffice. We also talked about the fact that yellow and blue pan traps might not capture the full range of what's there, and that maybe white ought to be added in. That seems to work well in Alaska. And some people had thoughts on how best to record flowers in each of the plots. As far as the sort of open questions at the end there, those bigger question ones, bigger picture ones, it was a general consensus that first and foremost, sort of the biogeography of the bees. We need to know what bees are where on BLM lands. And if we do that in a way that allows us to go back and resample those areas later, great. But that component of just figuring out who's out there is still a top priority. We went back and forth about whether that should be focused mostly on quote unquote pristine lands versus areas where different things are happening to the land. There's definitely benefits to both of those. We also talked about whether focusing on just a couple taxa might be an efficient and effective way to do this. Maybe just bumblebees, maybe just a certain couple groups. But the question is, do you focus on the bees that maybe are rarer, or do you focus on a really common bee like some species of Ceratina or an Agapostum and watch changes there because you can get so much data? We talked about nesting sites and whether that should be included, the potential for different nesting sites. And right at the end, does anyone know if Sam Droege has already looked to see how long it's beneficial to leave out bull traps? We thought there might be some unpublished data on that somewhere. So if anyone has a thought, please put that in the chat. I haven't found it. We also talked about the fact that the advantage to netting is that the bees are processed pretty quickly rather than sitting in a fridge somewhere for a long time, and that might be a component to highlight so that we could get to our data more quickly. So that was group 1. What about group 2? Does anyone know who group 2 was?
Group 2 was Hollis.
Oh, cool. Thanks. I didn't know. I think Rich is going to give our summary, but I can chime in too.
Okay. Yeah, I didn't really think too much about looking through the silent writing section of this. I don't know what we gleaned from that, but I'll just jump into the first question. That was the verbal discussion of sort of what pressing sampling technique or bee monitoring should we try to answer this year. We ended up talking a lot about logistics, just in terms of like human capacity, as well as spatial constraints. And funding constraints, and how we could possibly fit all of these, huge goals into these constraints that we have. We came up with some ideas about how to do that, picking some sort of close sites that are close to BLM headquarters that could potentially be used, where people could essentially do this out the back door as a good place to do this, or a way to trial that this year. We talked about whether it might be possible to trial some of the questions that Olivia mentioned during her talk. How long should we be netting? Seems like it's a testable thing that we could be sort of gathering if we gathered sort of the time that each bee was captured when we were processing during netting. I do think that Sam has run some of those numbers though, Olivia, so it would definitely be worth talking to him about. I don't know if he's done it with netting, but I do believe he has done it with bulls. Let's see, what else did we talk about? Yeah, then we just talked about the back end. So sampling seems to a certain degree to be the easier part of this, and then the processing and pinning and identification and data analysis part seems to be maybe we haven't talked about the logistics too much of that. There are already bottlenecks in terms of getting bees identified and long time lags, to say nothing about the wonderful expertise of the many people that are on this call that are doing those identifications. There just aren't enough of you all out there. And so how do we handle that? I guess the question really was, what is the sustainable number of samples and bees that can truly be processed? Yeah, I know, Jason, you and everybody else. So that was we ended up spending a lot of time talking about that. And then I think there's also just the bigger question here that is just sort of, what are really the specific questions that the BLM is trying to address with this protocol? Like, what is the bigger question? I think was a sort of big lingering question here as well. And then lastly, sort of how should the protocol be structured in order for funding constraints? We literally talked about this for the last 59 seconds as it was counting down. So we didn't get very far, but really quickly, we just recognized that the protocol that's developed needs to be sustainable, and maybe a last second idea that we should have a minimum number of sites that get monitored every year that is potentially expandable when funding is available, and or the resources at the back end in terms of storage of specimens and identification and all those things are available. So that's kind of where we got. Hollis, fill in any gaps that I may have left.
No, that's great. Those are all the main points.
Group 3 was Casey's group?
Yes, and I will be reporting out for group 3, and I haven't I don't feel like I have wrapped my mind around all of this yet to provide you with a good synthesis, but I'll do my best. We did do introductions, so we wanted to get to know each other, so that took a little bit more time at the start than we had for maybe the whole thing. But we talked, when we did our quiet writing assignment, we had a little bit of discussion at that afterwards. And I think there's some really interesting differences in the sampling unit. I think that's maybe the place where there's the most variability. I think everybody's done this and done it slightly differently. There's a lot of kind of broad similarities, I think, but the fine details, I think, are quite variable. And potentially, you know, we have a scalable system, kind of, I think, like Olivia talked about, where if we have more or less people, or, you know, we have more or less time, we could potentially do things slightly differently, as long as they kind of fit under this umbrella or this framework. So I think there's a lot to pull out of the sampling unit information that we input. And also, I think there's a lot in that last column of what other aspects of sampling should be considered. There's a variety of different topics that came up there that I think are going to be interesting to look through afterwards. But the one piece that we did discuss quite a bit after that table was kind of associated vegetation data, and what what we want to collect, how much time that takes, how valuable that is. The value of doing it in conjunction with AIM and having all of that detailed veg data, if possible, was definitely looked upon positively. Potentially talking about doing photo monitoring for vegetation as well, and spending more time back in the office to process that, especially if field time is limited, as certainly as it is in Alaska. We do that. So anyway, that was kind of the first chunk, and then we got into our other questions for discussion, and a few topics came up there. One that was kind of a new topic that I think is interesting is sanitizing nets to minimize pathogen spread. And I hadn't heard a lot of discussion about that in the bee world yet. Working in amphibians and in bats, that's a very common practice, but curious, maybe we can discuss that more as a RCN group at some point if there's a need or a risk there. And then I think the final topic that I think we're all aware of, but we hadn't really talked about today, is kind of the taxonomic bottleneck, again, kind of on the back end, and how much time we can spend, if we're out there collecting, millions of bees in the first couple years, who's identifying those and where are they all going? So that's the end.
Great. Brad's group was group 4.
Yeah, I'll report out for group 4, and for the sake of time, I'll be brief. We had a lot of really good input, and everyone thankfully added their input directly into the notes. So I'll just say that I think the notes touch on pretty much everything that was previously discussed, except maybe the sanitation protocol. A lot of our discussion was centered around development of the infrastructure, like the databases prior to like large scale deployment, and then making sure that we use a coordinated approach that incorporates parallel efforts and priorities because of the likely limited funding and availability of field crews to implement. So that's it. Short and sweet summary of the discussion.
Great, thanks. And Fred's group was group 5.
So I think you're going to be happy. All of the boxes are filled. Looks like everybody at the bidding part. So I'm not going to go over that. Interest of time. So group effort. Question 1. We really had the group sense of that. BLM really needs the baseline population inventory data before we can really start to answer a lot of the more advanced questions, like looking along latitudinal gradients and that sort of thing. So, it's a good, focus on that. Understanding the general species richness, habitat floral relationships, and then start on abundances. So that's the priority for now. Focus on gaps in undersampled areas and habitat types initially. We can't monitor what we don't know exists. Moving on to question 2. How should the protocol be structured? Had the suggestion maybe pairing sites and comparing different protocols either across similar sites or splitting a site, right? So that we can kind of tweak things and understand what's going on a little bit better. I thought that was a great comment. Again, start with undersampled areas, start with broad scale habitat, or start, focus either on undersampled areas, or there's another strategy could be potentially to focus on broad scale habitats that occur across all areas. Another suggestion about starting bi-weekly and then scaling up and down in terms of frequency, in terms of how much capacity and funding you get. Another approach could be to identify focal species and then scale up and down identification efforts, depending on the year and how much funding you have. Make sure our protocols are modular and hierarchical. That could help. Something not captured in there, but just in the general discussion, I think it's worth bringing up is sort of doing some data diving, in particular into the data sets that Olivia have. And I'm sure, Olivia, you're already doing this, but also maybe leveraging other people's data sets too, just to kind of answer questions about the protocols as they're being developed, right? Do you sample in the evening? Do you sample in the morning? That all of those kind of things, probably a lot of them can be answered by doing the data dive. And then also, part of that is everybody has opinions about certain things, and that data dive can really start to sort out what is opinion, what is not, right? Based on what data we got. And I'm scanning it, and that's basically what I got. Anybody want to jump in to add anything to that?
I'll just say one thing. Brian and I were messaging about this. I on your point about starting with inventories and getting a sense, like, that's definitely a growing, I would say, maybe consensus. It's a growing majority in these RCN workshops, especially our second one, which was focused on trying to understand what conservation practitioners need from monitoring data to make conservation decisions. There was one of the punchlines of that workshop, I would say, is the need to get a really fundamental sense of what is where. That came up in our group discussion too. And, a lot of the more sort of ecological monitoring type approaches, it makes sense to start more with getting a pretty fundamental sense of what bees are where. And from that, you can build a more effective monitoring program. So that's just a kind of a thing coming out of the RCN in general, I would say.
Thanks, Hollis. That's a wrap, guys. I think we got through everything we'd intended to today. I can stay on as long as needed if anyone has anything they want to talk about a little more. Brianna, I see, those are hand claps. I thought that was a hand up for a question. If anyone wants to stay on and share a thought they have, I'm happy to stay here and listen. But for those of you that have other pressing needs today, I want to thank you very much for your time and all of your incredible insights. You guys are super smart and given me pages and pages and pages to think about today. So I've got a lot to go over and a lot more questions actually now than I had when I started. So thanks again. Reach out to me if you have anything you think of later. I'll put my email in the chat one more time, and or reach out to the RCN and I'm sure they can pass it on to me as well. So here you go.